a word about precision medicine, which has become a big deal. And clearly with the president recently, Obama, making a commitment to precision medicine and giving out a bunch of money to give incentives, it's going to grow even faster. Why? And that is, it's very clear that it's the best medicine for people, and that is to focus on exactly what the problem is, limit treatments to drugs that do just what the problem is caused by. And so it's picked up at universities, and of course the focus initially has been cancer. We're coming up with the right approach is terribly important, both for the patient and also for the cost. And so that is moving rapidly in all fields, and it's wonderful the progress that's being made. So today the topic is precision medicine delivery on the promise, and we're going to hear about that. Mikael Dolsten is going to will moderate this group. Mikael was trained in both virology and immunology. He's an MD and PhD. Has been around the industry, has been in a number of companies, but currently he runs this gigantic research group at Pfizer, and is also an executive vice president, corporate type level, to oversee the entire portfolio. So he's controlling both what's done and what's brought in. And that's critical for any major company now, because you can't do all the research in the world. So Michael, I turn this over to you.
Thank you very much, Roy, for the kind introduction to the topic here. I'm very excited to be here, and welcome to all colleagues in the audience. I think it's a very timely opportunity with an esteemed panel next to me to discuss precision medicine and delivering on the promise. So you see next to me we have Jonathan Sheldon from Oracle, and of course a company representing health IT in a big sense. We have David Feltquate representing clinical Immuno-Oncology at BMS, a company that has been really moving this field for decades. Michel Sadelain, who is Director of Cell Engineering at Sloan Kettering and one of the pioneers in CAR T therapy. And a former colleague of mine, Phil Vickers, who is now head of R&D at Shire, is a company that has a lot of experience in rare disease. So I think this is a panel that covers so many key aspects of precision medicine today and in the future. I'll make a brief comment on the perspective we have at Pfizer and what we have done in precision medicine, and then each of the panelists will share a similar reflection from their business environment. So at Pfizer, we have had a strategy for a number of years to explore and embed precision medicine as a way to deliver pharmaceutical products with great innovative value for patients. And of course, we started, like many companies, exploring the wealth of data in oncology. And although it was part of our strategy, we were fortunate to make a real leap, as one of our drugs, Xalkori, was initially studied for c-Met lung cancers. Around 2007, publications in the Boston area in Japan indicated that a translocated gene, ALK, was driving 5-6% of lung cancer to in an oncogene addiction fashion. Interestingly enough, Xalkori did not just hit c-Met, but the structurally related ALK and also ROS genes. And our team rapidly expanded a cohort of ALK-positive lung cancers, which constitute, as you know, just a fraction of the lung cancers, 5 to 6%. And that allowed us to record immediately high response rate, durable responses, and led within 4 years to the approval 2011 of Xalkori as one of the first breakthrough drugs for precision medicine in lung cancer. I'm also proud to say that this year Xalkori was approved for a second indication, ROS, which is even more rare, 1 to 2% of lung cancers. And I think these two opportunities for ROS and ALK patients represent precision medicine at its peak. A drug like Xalkori would have been completely unsuccessful unless we used genetic diagnostic to select patients likely to respond. With that great experience in oncology, we have tried to push the boundaries of precision medicine into other diseases. And I just wanted to briefly mention the work we're doing with biotech partners in rare disease. Together with Spark Therapeutics, which is a spin-off from UPenn, we have embarked on looking at rare disease, and particularly monogenetic rare disease, to take on the challenge of correcting or replacing a missing gene that causes disease for patients. Initially with Spark, we focused on hemophilia B, a bleeding disorder. And you may have followed our progress, recent reports that we have now gene delivery with AAV, a well-known vector, into more than a handful of patients, and have shown that a single infusion of a synthetic factor IX gene with this virus can deliver a high and clinically very relevant concentration that allow the patients to seem to be independent of further transfusion of factor IX over several months. Now, this is early days, so we have to caution the optimistic data and of course, have more patients and longer observation time. But I think it underscores the opportunity for gene therapy in rare disease and precision medicine in a new approach. We're also trying to use precision medicine in more common disorders. And right now we're embarking, for example, in rheumatology to challenge the dogma of how much you can accomplish with today's biologics, which is very much treating all of RA patients in the same way. And we're looking at new ways to stratify RA with precision medicine, whether it relates to certain types of autoantibody that may distinguish subtypes of patients, or possibly also understanding some of the different immune infiltrate that you see in the joints. To move this precision medicine to its great promise, we need also new technologies that can allow us, instead of having quite invasive biopsy technologies, hopefully use more simple imaging and to integrate not just genomics data, but a broader data set from proteomics, epigenetics, and possibly even digital devices that allow us to monitor patients in real time and in a much more optimal way integrate molecular and phenotypic understanding of patients and what drug may really move the needle for each of them. I will just conclude this kind of more industry perspective of our optimism to go back what Roy alluded to, that there is a real simultaneous investment in the public domain, and we are very encouraged by the 1 million patient precision medicine initiative by President Obama. There is a 100,000 patient initiative by the Prime Minister in UK, focusing mainly on rare disease, cancer, and some special patient groups. And more recently, of course, the Blue Ribbon Panel, the Cancer Moonshot in US, hosted by Joe Biden, where I've been pleased to participate, that will look at new ways to expand precision medicine and move the entire field of targeted cancer therapy, particularly in the immunotherapy arena, look at new ways how we can understand these patients and deal with resistance mechanisms. So I hope this initial introduction gave you a high-level perspective of the opportunity, and I'll now ask each of my panelists to share some thoughts from their business environment, and I'll start with Jonathan here.
Thanks, Michael. So thanks for the opportunity to be on the panel. Since the topic is delivering on the promise, I wanted to talk a bit about the gap between good science and clinical application. And in some cases, the causes of this gap we can fix. In others, we can't for good scientific reasons, and I think we should probably be honest with ourselves in that respect. So let me start with what we can fix. So evidence. So in reality, no physician is going to change his practice behaviour without strong evidence of benefit. And one way for us to address that is clearly more clinical research that's focused on establishing clinical value and better health outcomes, which is really very different from establishing a sort of strong genetic association between a genotype and a phenotype, which is frankly what a lot of the research looks at the moment. I'd actually go one step further than that and say we should look at the financial implications of implementing these genetic tests into a clinical care pathway, not just the clinical outcome. That'll change the reimbursement landscape, which frankly is stifling the development and adoption of these precision medicine approaches, I believe. I mean, hand in hand with that, we need to develop acceptable evidence thresholds, which is particularly challenging because the extent of evidence required depends on the nature of the test and its use. And it won't be randomised clinical trials for everything, for sure. I think we're already seeing that. And genomic sequence data just makes it even more challenging to define those evidence thresholds. What we can also fix is implementation. So healthcare, well, in healthcare, implementation it's its own science, in effect. Each health system has its own set of peculiarities, if you like. It isn't a one-size-fits-all kind of problem, and it's very difficult to sort of generalise best practice. Probably what we can generalise, though, is that there's a lack of appropriate expertise amongst the point-of-care physicians and an associated shortfall, really, in access to medical genetics, genetic counsellors, molecular pathologists. I think what often gets missed, actually, in the implementation discussion is really the changing nature of the software needed. So, 5, 10 years ago, precision medicine was really one big research experiment. It was basically about trying to better understand the molecular basis of disease. It wasn't really going to affect patients a whole lot. You weren't actually going to make therapy decisions based on it. Now, clearly, that's changed a huge amount in the last 2 years. As we sort of usher in this new era of molecular decision support. And I think what comes with that is a need to transition our sort of thinking, our mindset, in terms of the way we look at the IT to support this area, to take more of a sort of enterprise-grade IT infrastructure approach. And that means we need to start caring about things, or caring more about things like security, scalability, auditability, how we interface with the EMR, how we interface with the clinical workflow. And I think, just in general, we've been really slow to realise that in the community, and I think that that is holding us back a little now. In terms of the gap and what we can't close, I mean, we can't forget the basics of disease pathogenesis. And, we know genetic associations in some cases have a small effect size, relative to contributions such as social and behavioural factors. Diabetes, heard this morning, it's a great example of that. And even if there is a strong genetic association, then it's still really difficult to change behaviours. And Michael referenced the Precision Medicine Initiative, and I think they very wisely broadened their definition of precision medicine, sort of past genomics, past omics in general, and more into self-reported data, remote patient monitoring, wearables. Fundamentally, how do you change behaviour? And really, the genomics is just one very small piece of the puzzle. You need that broader clinical context in order to change that behaviour. And such a broad definition places technology right at the centre. So it becomes a big sort of data aggregation and analytics exercise that, frankly, few large IT players can scale to, I believe. So just to close, there's been much in the press of late about precision medicine and population health management and how they're potentially at odds with each other. I actually think they're highly synergistic. I mean, precision medicine gives us the molecular tools to dissect the phenotype and the disease at a level of granularity where you can manage clinical and financial risk. That isn't to say you need to forget all the sort of foundational causes of ill health like poverty and obesity and education. It just means that you need to understand that, because precision medicine needs to work in that context if it's going to cross over to the mainstream.
Thank you, Jonathan. And I think this intersection of health, IT, big data, and medical and pharmaceutical sciences is a key area for us. So thank you for your remarks. Now I think we have an opportunity to move from big data to someone that is close to patients and do great science in a bedside medicine fashion. So, Michel, please share some of your thoughts on this area for us.
Okay, sure. Thank you. So Mikael has tasked me with telling you about the rise of T lymphocytes, that is to say T cells, as cancer drugs, and also to introduce to you a new class of molecules that a few years ago we called CARs, or chimeric antigen receptors. This fits perfectly under the large umbrella of precision medicine, but with a twist. So like all precision medicine, CAR therapy requires the identification of targets on the patient's tumor cells. However, in addition to that, it requires personalized manufacturing because we are rewiring the patient's own immune system to attack the cancer. And while some people have found this to be cumbersome because it crosses a line that certainly Big Pharma had not crossed until very recently, that is to say, making drugs from patients and their own cells. What has kept us and a few others excited about this is its potential to be curative, and I think that sets it apart from a certain number of other therapeutic approaches. So CAR therapy was born out of the conflation, if you like, of tumor immunology, advanced in scientific labs, and of cell therapy, and of synthetic biology and genetic engineering. 4 key fields that have come together with the perspective of generating from the patient's own cells a very powerful drug that is targeted very exquisitely to their tumor. So CARs, or chimeric antigen receptors, are synthetic receptors that retarget and repurpose a patient's own T cells. Some years ago, we showed, actually a few blocks from here, 30 blocks up north from here at Memorial Sloan Kettering Cancer Center, that we could design such receptors that recognize any antigen of your choice and instruct the T cell to attack the tumor, to overcome some of the resistance mechanisms that T cells may encounter in the tumor microenvironment, and extend the lifespan of those T cells. All of this is achieved through this synthetic receptor that again gives an instruction to the T cell. We had to identify a target to develop our program and to bring to the clinic, and we set our eyes on something called CD19, a molecule that's on the cell surface of a majority of lymphomas and leukemias. And then we undertook clinical trials after having provided the first proof of concept that this could certainly eliminate human tumors using human T cells implanted in a mouse. At about the same time, 3 centers came up with breakthrough results, all testing CD19 CAR therapy. At Children's Hospital of Philadelphia, or CHOP, they launched trials targeting pediatric acute lymphoblastic leukemia. At the NCI, the National Cancer Institute, investigators launched trials targeting lymphoma. And we at Memorial Sloan Kettering initially focused on adult acute lymphoblastic leukemias. As you may know, the results of that have been quite spectacular. Enrolling patients who have refractory disease, relapsed disease, lymphoma or leukemia, for which no drug was no longer available, or for which a bone marrow transplant would fail. A vast majority of those patients went into a complete remission, not just a complete remission, but what's called a molecular remission. And so this was not achieved by giving a chemical. It was not achieved by giving an antibody. It was achieved by engineering a patient's own lymphocyte and infusing it back to the donor. So the FDA gave breakthrough designation to these studies at Sloan Kettering and at CHOP. And today there are 3 pivotal trials that are underway, seeking hopeful approval for these drugs, perhaps next year. Novartis has sponsored a trial in pediatric ALL, KITE, again in lymphoma, and Juno Therapeutics, a trial in adult ALL. So I'll leave that at this as the introduction to this particular aspect of precision medicine, and I think later we'll get to what the challenges and perspectives are to make this more broadly available for more patients and more cancers.
Thank you, Michel. And from that optimistic perspective of close to curative aspiring responses with cell engineering, I'm excited to ask David Feltquate share his thoughts and learnings from the quite dramatic experience also with the biologicals in the field of checkpoint antibodies in cancer.
Good morning. Thank you for the opportunity to speak. So I'm going to talk to you about a different form of immune therapy, but it falls under the same bucket of using the immune system to target our tumors. We are in the midst of a revolution in the areas of oncology drug development and treatment. Breakthroughs in our understanding of how tumors evade the recognition and destruction by the immune system have launched these breakthroughs, and it's now transforming oncology. And as we start to learn more about what these mechanisms are and how to target them, we can expect even more progress. Today, there are two types of immuno-oncology medicines that fall under the class of checkpoint inhibitors. One of them is an anti-CTLA-4 medicine, and the other is a group of medicines approved that target the PD-1 and PD-L1 pathways. I'm not going to go into the exact biology of how these work, but suffice it to say that what they do is they are a countermeasure to what the tumors are doing to prevent the recognition and destruction by the immune system. And we've been very successful. With these two types of medicines, in total there are 4 medicines now approved. We have shown that they are efficacious in extending survival in 6 malignancies, actually survival in 5 malignancies, but there are approvals in 6 malignancies, spanning both solid tumors and hematologic malignancies. So the potential is that this is a very has broad implications in terms of how it can be used in oncology. And some of these, there's been one attempt thus far, or one successful attempt of combining these two types of medicines. There's an approval in melanoma in doing that, but there's signals of activity that these combinations can even have the potential better activity than either of these agents alone in a variety of other tumors. And so this is the tip of the iceberg. These are just 2 targets. There are over 100 different types of drug candidates under evaluation right now in this area, targeting at least 30 different distinct mechanisms, some of which we may ultimately learn are key in helping the immune system turn the tide and recognize and destroy cancers. You talked about optimism. We're very optimistic about how active and how much this field will transform. Now, this is a panel devoted to precision medicine, so I want to spend a little time talking about that. What's great about all these new kinds of targets is we are approaching the development with the aim of tailoring these treatments towards identifying where these targets are most active. So the recent example is for PD-1 pathway drugs, that in many cases, the patients that are more likely to benefit from these treatments have tumors that have evidence of expression of markers of that pathway. It's not a perfect marker. There are patients whose tumors are not expressing this pathway that also respond to these treatments. But this is an indicator of where the field will go. And as we develop new kinds of treatments, new pathways, I can guarantee you that what will be examined are those specific expression of those pathways as the actual predictors for who may benefit and who may not benefit. So we know now that single agents can help treat a number of patients, but it's the minority of patients. So combinations of treatments are clearly going to be where the field will go. Doublets, triplets, potentially even 4 drugs targeting distinct pathways. And it is the hope as we make progress, we will be able to molecularly probe each cancer and tailor specific cocktails for each individual patient. And if we're able to do that, I expect in the future the field of oncology will look very similar to what the field of infectious disease now looks like, where we're able to create these kinds of regimens and hopefully potentially eradicate most people's cancers. Thank you.
Thank you very much. And after 2 leading scientists discussing oncology, I'm pleased to have someone bring us back to rare disease, Phil.
Thanks. Good morning, everybody. Thank you, Michael, for inviting me to speak. So what's Shire's thinking around precision medicine? I would say, we are a company that is focused on being a leading company targeting rare and specialty conditions. And so precision medicine needs to be at the very heart of our thinking. Often what we are doing is targeting genetic diseases and thinking about what is the exquisite way of targeting that small population that suffer from what in some cases are really terrible diseases, while recognizing if you are exquisite in targeting those therapies, it's a different beast, because you cannot use those therapies in some cases for larger populations. You're really restricting yourself in that targeting to this very specific kind of population. But we do that, and the way that we do it is to try and understand the genetic basis for disease, and then we'll look at what is the best way of targeting those individuals. And in some cases we will move forward, or are moving forward, gene therapies, for example, for haemophilia, mRNA replacement therapies for mutations in the gene where you make less mRNA or it's mutated. We will try and provide normal mRNA to patients. So for cystic fibrosis, for example, we might target in that way. Protein replacement therapies, so parathyroid hormone for hypoparathyroidism, enzyme replacement therapies such as elaprase for Hunter syndrome. So really very, very targeted. And there are some benefits as an R&D organization to doing that. If you target a population that is very well defined, then you can have more of a homogeneous population in your clinical trials. So you may hope that the responses you get will reflect that, rather than having a variety of patients, some of whom respond, some of whom do not respond. Also, if you know that a patient is not making an enzyme, and you can provide them with that enzyme at the right place and at the right time and at the right level, your potential probability of success for such a program may be higher than a small molecule that might have off-target effects or toxicities associated with their mechanism. So there are some advantages, but there are some very clear challenges associated with doing this kind of approach, and perhaps later we'll be able to talk about some of those challenges that you face in the rare disease space, because the way that we look at it is, Michael gave some examples in cancer, for example, where now you can define a population where years ago you could not do that. And I guess we look at that and think that down the road you can actually look probably at more common indications really being a mixture of a subset of rare diseases. And so I think some of the learning that we have in how to address rare diseases will apply to those subsets. And even companies that are now focused on rare diseases, it gives them an opportunity to broaden their focus to attack more common indications that you can break down in that way.
Thank you very much. So I wanted to ask each of the panelists one particular follow-up questions, and starting with Jonathan here. Tremendous amount of data through new technologies from genomics to digital devices, and you did touch upon the clinicians and the EMRs, how to on one hand make them user-friendly, and on the other hand how do you create the continuity from the practicing physician to the scientific explosion. There are some initiatives by NIH. Accelerating Medicine Partnership is one, to create a knowledge book for diabetes to understand its different subtypes of disease. But I'm a bit concerned that we will see a lot of new science evolve, but it will do it in a very fragmented manner. Whether we look at the EMR system in the US, or if we look at the revolution in Immuno-Oncology. So what do you think could help us to bring the knowledge together that could allow each of us to faster translate, whether we are the scientist, the pharmaceutical company, or the physician for the patient?
Right, so I mean, our philosophy at Oracle is to aggregate once to power the sort of multitude of analytical applications that are needed to address the kinds of questions that Michael mentioned, either in the cloud or on premise. It really doesn't matter. So we're aiming to create a sort of single integrated view across what is a very heterogeneous data landscape, both in terms of phenotypic and genotypic information, along with financial information that relates to the cost of care. And I think that's becoming increasingly important with the new macro legislation, for example, and this whole sort of shift in healthcare to value from volume. So, specifically, I'm talking about being able to integrate data coming from the EMRs like Cerner and Epic, maybe from clinical trial systems like Inform cloud service, clinical and research lab systems, registries, image files, increasingly now remote patient monitoring devices, wearables. And then, of course, there's the omics piece. So you need to be able to handle all the different flavors of genomics, whether that be gene panels, whole exome, whole genome, RNA-seq. And as Michael said, it's a huge amount of data, and I'm sure we can't even begin to imagine the amount of data we'll be managing around the patient in the next 5 to 10 years, particularly as some of the newer omics technologies come on board. So I do think scalability and just ensuring what you use today will be fit for purpose next year, so sort of future-proofing, if you like, is going to become increasingly important. And we invest a lot of time at Oracle, frankly, at testing these kinds of things. And I think what works for an individual project will not work on an enterprise scale, and it's really important to acknowledge that. And I think as a community we've been slow to do that, as I said in my opening remarks, and I think that's holding us back. The other point I would say, and I really can't emphasise this from all the projects we've worked on, is data quality is absolutely essential. So if you get the data governance piece wrong, the value of the analysis, the interpretation, erodes away very rapidly. It's important to be able to standardise, normalise the data, make it consistent in terms of terminologies and measurements. The bottom line is all that dull stuff, that's what the good science sits on, and we aim to take care of that. So it's equally important from a clinical as well as an omics perspective, and if anything, the most difficult part is the healthcare data, because most of the standards there are only really partially adopted. You have LOINC, you have RxNorm, ICD-9, ICD-10. There's a lot of challenge there. If you look at value lists like height, age, weight, blood pressure, there are no real standards for that data in healthcare, and that makes the integration challenge difficult. Thanks to the bioinformatics community, we now have some reasonable standards in genomics. And as Michael alluded to, there's a number of efforts now that are tuning those standards to make them more relevant to the clinic. So you are in a position then to push this information back into the EMR and back into the clinical workflow. I mean, our goal is to get the data into an analytics-ready form for whatever application or use of that data follows. So that may be things that we build, it may be applications that our partners build, it may be in-house applications, maybe open source applications. And the reason that is so important is best practice is clearly a way away at the moment in precision medicine, and we don't want to force anybody down a particular analytics strategy. We really want to support the application, the analytics ecosystem with good quality data. And I think it's really important to remember that the sad reality is that most of these projects fail because of data access and data aggregation. They don't get as far as the actual analytics step. It's actually very difficult to get the data in the first place and then to standardise it and aggregate it in a way that's meaningful. So if you look at that in another way, there's actually plenty of good analytics out there today that never gets to see good data. Michael did ask me prior to just give a specific example, so I thought I'd mention Penn Medicine. It was mentioned earlier as well. So we're currently managing about 3.2 million patients there. The data that we're ingesting comes from their clinical trial management system, from their Epic installation, from various clinical and lab research systems. There's a registry, there's a biobank. In terms of the genomics, there are tens of thousands of patients now with gene panel, whole exome, whole genome. And, what's important about this is from a scientific perspective, it enables them to ask questions of that data that would have taken months to ask those questions in in minutes. And for science, that means you actually ask the question. If you're going to have to wait months for your answer, you forgot why you asked the question, you go off and do something else. So it actually changes the way you approach your research, and enables them to ask questions like, find me all the lung cancer cases that have a EGFR-activated mutation that failed primary EGFR therapy, that have gone on to develop new metastatic disease. So quite a complex query, but very difficult to answer today in reality, because you're pulling data from a whole variety of different source systems.
Thank you very much. So Michel Sadelain brought us into the new modality of engineered cells, and you heard this transformative outcome in very advanced lymphatic leukemia and also lymphoma patients. And I think his group has tried to further refine these CARs, and I think you hear them speak about armed CARs to be resistant to the hostile environment in certain cancer. So the question is for us, and to have Michel address, I think, is this going to be a tremendous achievement in a niche but important disease like lymphatic leukemias and lymphomas, or is there a more universal role for cell-engineered therapies in cancer, possibly going broad in blood malignancies? And do you think we should have some optimism, and by when will data support that potential optimism, that it could be a modality going beyond blood cancers in some of the difficult to treat solid tumors?
Okay, great questions, Mikael. CD19 CAR therapy has established the feasibility of engineering patients' own cells for reinfusion, and of course its efficacy in B-cell malignancies, as you pointed out. And this has led, this has resulted in several big pharmaceuticals, Novartis being the first, Pfizer now and others to pursue this approach that till now had not been approached in the realm of big pharma. And several biotech startups also have formed in recent years to take on this challenge. And so the questions are exactly what you said, is can this work for other cancers and solid tumors in particular? And can we make this more broadly applicable? Because there is a bottleneck here in the requirement for patient-specific manufacturing. So the future directions really for this field focus on three levels. First is the identification of suitable targets for these CARs. If we have a target, we can build the CAR for that target, but we need to identify those. So here we're exactly in the realm of precision medicine, having to search for more such targets. These T cells are very powerful. They will not just destroy the tumor that expresses the antigen. They can also attack a normal tissue that expresses the antigen. And that has occurred in some clinical trials. So we know that we need to find targets that are highly restricted to the tumor. So there are big efforts underway to do that. We are in particular focusing on AML, myeloblastic leukemia, and glioblastoma, and many groups are looking for targets in a wide range of tumors. Again, because this approach is in principle applicable to many, if not all, cancers. Secondly is to make more powerful T cells. We're fully aware that we need to overcome a very immunosuppressive microenvironment in a number of tumors. So the T cells that reach the tumor have to overcome those mechanisms. And there's a number of exciting approaches that are underway. For this reason, I think that the CARs as they exist today that are so effective in lymphoma or acute leukemias may not work just as such in solid tumors, but there are in laboratories and now entering trials a number of strategies where the CAR T cell not only kills the tumor but alters the microenvironment. And this goes to the essence of using a cell for therapy. The cell can achieve multiple tasks. It can secrete proteins either constitutively or in inducible fashion, and it becomes what we sometimes call a micropharmacy. Think about a T cell that's been infused in blood, has migrated to the tumor, and now can release in the immediate vicinity of the tumor a number of compounds, natural compounds or synthetic compounds. And I think this is very promising and will probably unfold in coming years. At Memorial, we have trials in ovarian cancer, breast cancer, prostate cancer that next year will open, testing this concept of this CAR T cell that's used as a device to alter the microenvironment and recruit more T cells against the tumor. And the third direction for research is to improve the manufacturing of T cells. And there are two visions. One is the autologous setting, for manufacturing for every patient. Until now, this field has developed in academia, but for the past few years, we've seen now many companies taking on the challenge. And I think manufacturing will be dramatically altered in the years to come. You know, robots, chips, miniaturization, cutting out the technicians and fellows that today are still needed to handle this production. I'm convinced there will be a lot of progress. And then there's a second approach, which says that you can make cells from one donor, but not just for one recipient, but for multiple recipients. This is very exciting. This would, again, bring these cell therapies out of the mold, if you like, of autologous approaches. In fact, Pfizer is a leader in this, planning trials that will test T cells that can be administered to multiple recipients. And here, too, we have now a collaborative research agreement with a group called Fate Therapeutics that's very expert in pluripotent stem cells. And the question that we're asking today is, can we actually bypass the donor? Can we actually make these cells in a dish, derive them from pluripotent stem cells, and from those cells generate cells that have been genetically engineered with a number of optimized properties? And I think that's, of course, one of the futuristic directions for this field.
That was a fascinating introduction into the future. I heard about synthetic man-made cells acting as your local micropharmacy, and I'm hoping there is a remote control to these pharmacies.
Yes, there are. There are drugs that can be used to turn on and turn off genes, so the clinician can have control over the activity of those T cells. There are fail-safe mechanisms. These cells being so potent, there are instances where it may be necessary to recall the drug, if you like, inside the body of the patient. And there's really a tantalizing array of approaches right now. In particular, and this is precision medicine as well, using gene editing, so CRISPR-Cas9 and other approaches to introduce genes, to delete genes or introduce genes into cells that will come to express their gene at the right time and at the right moment. And again, this is this conflation of genetic engineering and synthetic biology in human cells.
Thank you. So inspired a little bit by these high-tech therapies coming out to David, and of course the transformative aspect of durable response with checkpoint inhibitors that David spoke to. One of the challenges that I'd love to have David share his thoughts on is that to make that more optimal, on one hand we need to do the tumor sequencing, understand the potential oncogene to treat with traditional drugs. We heard about introduction of immune markers like PD-L1, but we also know the mutational load of the tumors. We heard about suppressive element in the hostile tumor environment. So David, to really see this field of checkpoint combination flourish, how much do you think we need to push the boundaries of multi-marker analysis of the complex tumor? And is it static, or do we need to think about a real-time monitoring over a therapy journey of a patient?
Yeah, it's challenging to know how this is all going to evolve. You asked about checkpoint inhibitors, but I think we need to really think more broadly around immuno-oncology, all the different pathways that may be employed to target cancer. I'm not going to project to the future. I'm going to actually talk about a few examples that are going on right now, just to give a feel for where this may go. So I mentioned earlier around PD-L1, measuring that protein within a tumor, on tumor cells, on immune cells, as a marker. And recently, earlier this week, actually, the FDA approved a PD-1 agent for the use in patients with non-small cell lung cancer that have the presence of this marker in 50% of their cells. The data is outstanding. It worked better than platinum doublet chemotherapy, which has been the standard for over 20 years. So that's transformative. When you look at responses, and the experience and for that drug was it also improved survival. But if you look at responses, that single agent treatment led to responses in about 45% of patients, as compared to platinum doublet chemotherapy, which was in the order of about 30%. But that still is a little more than the majority are not having full responses to this treatment. So what we're starting to see, though, is that I mentioned before about combinations. It's possible that we can use that as a marker to enrich for a group that then may respond to multiple treatments. So at ASCO this year, there was some data published, a small data set, but it was very intriguing data combining what I was telling you about before, two different types of checkpoint inhibitors. And in that exact same population of 50% expressors, there was over 90% of patients who responded. And a number of them had complete responses. In fact, some of them were pathologic complete responses. What that means is someone went back in and actually did surgery, remove what they thought was tumor, and they found there was no tumor there. It was just scar. Those patients have a high probability of being cured. So think about that. In this subset of patients in non-small cell lung cancer, the most common cancer worldwide, there was a subgroup that you could identify with a marker, and almost 100%, so it was greater than 90%, responded, of which a portion of them may have been cured. That is unprecedented. Now, that obviously needs to be tested in randomized studies. Those studies are ongoing. But that gives you, it sheds some light on, I think, how we're going to stepwise approach this field. You have a marker. You show improvement. If it's not 100%, because that is what our goal is. We want cure for all of our patients. You then have a focused subset to benchmark, to improve upon. The next question then is, well, what do you do about the patients who aren't at that 50%? What are the ones that are positive but they're not high? Well, again, you have now another benchmark. And this is where you may need to have 2, maybe 3 drugs targeting distinct pathways to improve there. This is going to be how we're going to stepwise improve care for all patients. Now, there's another example, completely different kinds of biology. There are patients that harbor tumors harbor abnormalities in certain DNA repair genes. We actually, keeping the fidelity of our DNA is so important. We have over 100 genes devoted to repairing our DNA. A small group of those genes do something called mismatch repair. And if you have an abnormality in them, you don't fix mutations that are occurring. You will generate millions of mutations. These then are visible to the immune system. And so patients whose tumors have these abnormalities, they are highly visible to the immune system. And as such, tumors that evolve in that environment, that actually survive, that didn't get eradicated by the immune system, the tumors are employing mechanisms to keep the immune system at bay. And it turns out the PD-1 pathway is probably a key component of that immune evasion mechanism. And we have seen now in the last year and a half, that if you use a PD-1 drug, patients who would not respond or would have a very, very low response rate to chemotherapy, maybe on the order of about 10%, less than 10%. We're seeing responses of 30, 40, 50%, and these responses are quite durable. Again, it's not the majority. So what is needed is now adding combinations. And we saw last year at ASCO, when you add 2 different types of checkpoint inhibitors, you get an incremental improvement on that. And I think, again, this is exactly what is going to be happening as we start to develop different kinds, even going beyond checkpoint inhibitors. We will have markers that will start to subdivide, and we will basically master, I think, how to manage each of these patients, one subgroup at a time.
Thank you. So coming back to Phil, I wanted to ask, where do you think some of these tremendous learnings that end up as headlines almost weekly in medical journals about breakthrough in oncology and highly specialized disease areas can be translated to more common diseases where the treatment options haven't accelerated to the same pace. For those of you that read Global Burden of Disease that was released recently in Lancet, it indicated that the largest disease burden globally, when you look at aggregated diseases, may be in neuropsychiatric, from neurological to psychiatric to dementias. And it's an area where we have, put a lot of scientific investments, but of course we're still far away from this rapid pace as we heard in cancer. So Phil, your company looking at rare disease, how do you see the opportunity to translate some of those insights? And you have worked, I think, on rare neurological disease to some extent, and do you think that could accelerate some of these common disorders?
I certainly do. And I think there's tremendous opportunity there, but it is not going to be straightforward. I think we are fooling ourselves if we think, well, we'll have a large indication, we'll just subset it into these smaller indications which might have a very defined genetic background, and then we'll just target those. There's obviously tremendous challenges associated with that. You're right, we do have approaches for neurodegenerative disorders, CNS disorders. Maybe one I'll give you an example of would be metachromatic leukodystrophy. For those people who may have watched the CBS Sunday News last Sunday, there was a piece about MLD. It's a terrible disease which we are targeting. And the way that we are targeting this, well, the cause of the disease is a mutation in the gene for what makes an enzyme called aryl sulfatase A. And if you have a mutation in that gene, which then makes the protein ineffective, you get a buildup of what are called sulfatides. These are toxic materials. They cause demyelination of neurons. And so our precision in trying to address this disease is to provide those patients with the enzyme. We take very great care in the enzyme, but it's to provide that enzyme to those patients. And then the second element of the targeting is if you just give an enzyme intravenously, it will not cross the blood-brain barrier. So we had to design a device that would allow us to provide the enzyme directly into the CNS, an intrathecal device, so you could put the enzyme directly into the CNS. So we're targeting it on a molecular basis, but also on a delivery basis. And that is not a trivial thing to do, obviously. If you're putting hundreds of milligrams of protein into the CNS, you could get aggregation, for example, of proteins. But on the face of it, it might appear that that's relatively straightforward. A patient doesn't make an enzyme, we provide the enzyme, and you can address the issues. But it really is not that simple. And some of the challenges we have had to face in that program would be relevant if you're subsetting patients down into more genetically defined populations within a large indication. You can get varying mutations. You can get lots of mutations in the same gene. And in metachromatic leukodystrophy, you can get an infantile form of the disease, very severe form where the patients basically do not make the enzyme. They get a very rapid buildup of sulfatides. Children are bedridden and frequently will die before the age of 6. You can get other mutations, or you get a juvenile form of the disease where they may make some of the enzyme but not all of the enzyme. And so you get an intermediary type of disease. And then you may get a milder form of disease due to other mutations, which is the adult form of the disease. Now, if you think about that, you get 3, it's all metachromatic leukodystrophy, as if you're getting a mutation in a broader population which is defined. However, the clinical manifestation of infantile, juvenile, and adult is very different. So what those patients need is very different. The course of disease is very different. If you have a very slowly progressing adult form of the disease, then how can you even run a clinical trial? Very small population, very slowly progressing. And the medical need is in the infantile population. Medical need all over, but the highest medical need is in the infantile population. So that's where we want to focus our efforts. But you think about this from a research perspective. As you go into looking at these subpopulations, there's very little published. So how do you run a clinical study? What are the endpoints that you need to run? You shouldn't assume just because it's a well-defined population that they will respond in the same way that a broader population will respond, and that clearly is not the case within MLD. So understanding and partnering with patient associations is going to be increasingly important to understand the natural history of disease. And in MLD, as in other diseases, we actually run clinical trials that are just natural history studies just to track how those patients go with time. A great opportunity for public-private partnership in that kind of an area. Newborn screening would be a tremendous advantage, and another area that we're very interested in. As you go to clinical, as you subset down, simply finding patients for clinical studies is incredibly difficult, and so we have to go out with social media through third parties to try and make people aware of the clinical trials, and do you have these characteristics so that you can be enrolled in the study. The point around changing the way that you run your clinical studies. Can you have better ways of measuring cognitive ability rather than doing an IQ test once a year? Can you have technologies which are appropriate for regulatory authorities, where with current computers that you could monitor the neurocognitive ability of children on a daily basis, rather than having to do 6-month or 1-year cognitive ability tests. Regulators have never seen drugs developed for a disease, so early engagement of regulators is extremely important. Medical is very important, so I think the whole lens through which you need to look at drug development.
Thank you. Thank you very much, Phil. An opportunity now for questions from the audience. I know Nicholas Dracopoli expressed interest here. I don't know if you have arrived yet, but you would be given the first opportunity. Otherwise, anyone that has a hand available, please welcome we have Matt Harper here.
Hi, Michael. So particularly with the tyrosine kinase space in oncology, a lot of the best drugs, including Xalkori, including Gleevec, have been surprises. But we're in an environment where finding patients for trials and testing drugs is very difficult and takes a lot of resources, where we might be better off getting 1,000 flowers to bloom. How do we lower the cost of development so that we can test those drugs in more patients, especially when you talk about ROS1 or ALK, you're going to need a new drug for each of the mutations that cause resistance in those patients. And so you're going to go from 1% to 0.5% to 0.25%. How do we make that feasible when people are complaining about the cost of drugs now?
Yeah, obviously it's a key theme to accelerate drug development, and I think a part of it you heard today, to embark on more breakthrough medicines where the magnitude of medical advance is much more dramatic. Sometimes it's due to a new modality. We heard about CAR-T, checkpoint inhibitors, gene therapy today, but it could also be careful selection of a patient group. And when we are able to make those dramatic medical advances, it allows us on small patient numbers to bring a patient forward into a registration type of trial. And I do think that FDA has been one of the leaders through the breakthrough designation to stimulate industry and collaborate with them to advance the field. In Europe, we have the PRIME designation that is similar, and also in Japan. So under certain aspects of drug development, I think we all are very optimistic. What is still the daunting challenge is for these large unexplored areas, when we mention neuroscience, but we also see new technologies, imaging-based, much more sophisticated psychometric tools using digitalized technologies. So I hope we'll also see these type of dramatic responses in some of these diseases that would allow companies to work with regulators and make journeys that used to be 10 years into 3-4 years. And possibly, the ability to follow patients on breakthrough drugs that are approved using electronic medical records, if we can integrate them across our nation and in many other countries, that would allow the boundary between clinical trial and medical practice to be more a continuum rather than a wall. So, Matt, we recognize the challenge, and we hope, if we're sitting here 5 years from now, that the statistics and metrics would look different. Any other question here?
Yeah, this is Balu from Pharma Innovation. Quick comment, if you could comment on neoantigen vaccine as a booster to some of the discussions you had here of the checkpoint inhibitors as a combination. Is that something to be considered?
Sure, I'd be glad to comment. So the question was the use of vaccines based off of characterizing neoantigens from tumors. Yeah, there's actually a lot of interest in this, and I know that there are a number of startup companies and others that are looking at this. This is going to be a challenge, and I want to take a step back and put things into context. So for about 30 years, the development of immuno-oncology was stifled because there was a belief that the key problem was that the immune system was not recognizing cancers because cancers came from self. And so for 30 years, there was an effort to come up with vaccines. And for 30 years, there was really no success. It was when we had a better understanding of the immune system, evasion mechanisms, and we started to create tools to target that we actually saw the success. And so what we learned something very important. That fundamental hypothesis or the fundamental assumption was wrong. The immune system can see the tumors in probably almost all instances. It's the challenge is those evasion mechanisms. Now, despite that, is it still possible that vaccines, and in particular ones that are tailored based on neoantigens, what actually is being produced by tumors, can they be of value? And yeah, I bet we will find that there in some cases they will be, but I don't think that they're going to be the panacea for a large amount of the work that we're doing.
Thank you for sharing your thoughts. And I do think, having taken part in the Blue Ribbon panel for the Cancer Moonshot, that we'll probably see a reignition of this area of cancer vaccines as we're able to break this functional tolerance in the tumor. And I do think the question was posed on a very important intersection of these new drugs that can take off some of the suppression and could potentially lead to revitalization for cancer vaccines when combined. Any other question here?
Mikael, you mentioned the vision to cut back the time from bench to bedside. You mentioned 4 years, maybe even 2 years. So one thing that would be required for that would be conditional approval. So what would we have to do as an industry, working with regulators, to make conditional approvals more the rule than an exception?
Yeah, the rules rather than... I do think where we have been touching upon is the key theme. Regulators need to balance benefit versus risk. When the therapeutic advance is dramatic, when you have more than 80% response by CAR-T in certain lymphatic leukemias, or if you see in rare disease, dramatic responses, we have seen the regulators being willing to approve drugs on just tens of patients or up to maybe 100, 150 patients. So I do think that's really where it's going to make a difference. The oncology division has been really a pioneer, and I think the leader of that division has had tremendous courage to, challenge the system. So I think for us to being able to engage other divisions of FDA and find the right ways to be able to monitor drugs in real time. And I think Oracle and other providers, if they can unify the medical records, that could allow this important balance between benefit and risk be extended into more areas. I welcome one more question. That's why I kind of took a quick bite at this.
Yeah, so
The last question here.
my question's in the immuno-oncology space. So when we look at PD-1, PD-L1, patients with high mutation burden are typically the ones that show the best response. As we begin to apply external pressures in the form of therapeutics in an environment like that, we're really starting to think about antimicrobial resistance mechanisms, right? So we've got unstable genomes. We've seen some literature emerging of patients now with resistance mutations. So two questions. One, what are the ongoing R&D activities to try and catch that ahead of time? Because we know that the system is set up that way. And then two, from a diagnostic perspective, IHC has really no ability to identify that. So what are the diagnostics that need to come out to be able to support really capturing resistance as it evolves in these patients from an ongoing monitoring perspective?
I think there are a couple of questions within the broader question. So number one, although there is an association with mutational load and general responsiveness as clustered by different tumor types, more and more data is coming out that is really not a good arbiter of who will benefit or will not benefit from checkpoint inhibitors. So case in point, renal cell carcinoma. Renal cell carcinoma clusters around the low to intermediate as a cluster of tumors. Yeah, we've seen some of the best activity in that tumor type. And there's emerging data that it might be shared antigens actually that is one of the drivers for that. But I think you were thinking more around, can we identify markers of resistance that might be part of that? And I think there's some interesting data that's coming out around what are these things that are driving resistance and are they targetable? So Tony Ribas published a great case series a few months ago. A couple of individual patients with melanoma who were receiving checkpoint blockade. And he was able to show these are patients who responded. But he was getting serial biopsies, and eventually they had relapse, or they had one of the sites of disease relapse, but others not. And he was able to molecularly probe what was happening. And he found a couple of different explanations. In one patient, he found that the machinery that processes and presents antigen on the cells, they call this the MHC apparatus, there was a defect. And so in effect, the tumors that grew out were the immune system was blind to them. He also found that there was a pathway, the JAK pathway was activated in a couple of other patients. So in where pathways are activated, we probably are able to drug these. And the question then will be, can we have known way back at the beginning that there was a subpopulation that we should have known about and then intervene? In terms of loss of antigen presenting machinery, that's going to be a tougher challenge. It's almost like tumor suppressor genes. When you lose something, what do? The good news is the immune system was smart, and over the hundreds of millions of years while it developed, it figured how to do that, and that was natural killer cells. Natural killer cells actually work in part by recognizing cells that are not utilizing this kind of machinery. And so it's possible that maybe some drugs that are in development right now to augment natural killer cell activity might be one of the ways we do that. I think it's going to be a challenge, though, again, to go back and find out who those people are that have the subclones that come out. But you know, the technology is amazing, right? Who knows what we'll be able to actually ascertain in the future.
Thank you very much. We are heading down to the wire here on time. And I just wanted to stimulate the coffee break. And you know, one suggestion for you to go to the coffee break and think that if we were to have this panel by 2020, which of the panelists will speak about cancers that we are able to cure? Where will we hear about cancer patient counting survival in 2 decades? Which of the rare diseases will have gene therapy as standard of care? And would I invite a neuroscientist representative because of a dramatic advance in Alzheimer's? So please take the coffee break, speak about it, and let me know for the future. Okay. Next coffee break in the afternoon.